Okay, hi, my name is Elizabeth White, and today I'm a second year biology and chemistry AOC at New College of Florida, and today I'm going to be talking about the research I've been doing over the past year on the differentiating morphological characteristics between Tillandsia fasciculata and Tillandsia utriculata seedlings. So these are pictures of the two species that I just mentioned. One of them's a Tillandsia fasciculata and one's a Tillandsia utriculata. So based on these pictures, do you think that you would be able to tell them apart, and how? Yeah? Yeah? How?
Number of leaves.
Number of leaves, okay.
Overall shape.
Overall shape, yeah.
Trichome density.
Trichome density, yeah. All good points. But yeah, no, not easily. Yeah, you're right.
You have thousands growing together.
Exactly. Yeah. So the differences, although you can tell clear differences between these two, they're definitely a lot more nuanced than the differences as the adult forms. So on the left is Tillandsia of flowering Tillandsia fasciculata, also known as a cardinal air plant because of its short red inflorescence in comparison to Tillandsia utriculata, which is on the right, that has a much larger just green inflorescence that can grow up to 5 feet, which giving its name the giant air plant. And as you can also see, the growth forms of the two are also different, being that Tillandsia fasciculata, the leaves are a lot slimmer and more pointed in comparison to Tillandsia utriculata, or the giant air plant, which has wider and more curled underleaves. There's also a difference in color. The Tillandsia fasciculatas are generally more gray and lighter than the more bright green utriculatas, especially towards the leaf base. But it takes a really long time for these differentiating characteristics to show up. So in the Selby greenhouses, I had access to seedlings from around 5 months old to around 3 years old that I was able to take measurements of and develop this growth chart. And basically what this is showing is that over that 3-year period, there was only growth of around 20 to 25 millimeters. And like I mentioned before, these plants can get up to like 5 feet tall with their inflorescences. So what this is showing is that a lot of these plants' lives are spent as seedlings and without a lot of really distinguishing characteristics. So before I get into my methods and what I was doing, I just want to give like a little bit of a background on what these plants are and what unifies them. So they're both in the genus Tillandsia, which is in the family Bromeliaceae, and they're both considered epiphytic, meaning that they grow on other organisms and obtain their nutrients and water from precipitation or humidity or just general environmental factors. And to do that, these two plants have characteristic trichomes, which are basically what makes the leaves look fuzzy, and they're modified plant hairs that there are a couple of different theories of what they could be used for. One talking about how that could be the means that they gain moisture and nutrients from the air and from precipitation. Another that this is how they retain moisture. Yeah. So in recent years, the Mexican bromeliad weevil Metamacius calizona has caused a rapid decline in population numbers of both of these species. And basically what happens is that the larval form will eat out the bud of the plant, causing the leaves to separate from the root core and causing the plant to fall on the ground and therefore die. So this is a lot bigger of an issue in Tillandsia utriculatas, being that the way that they reproduce, they have to go all the way to seed before they can reproduce at all. So if the weevil intercepts them at any point in that process and kills them before they're able to go all the way to seed, that's it. They can't reproduce at all. As opposed to Tillandsia fasciculatas that are able to reproduce by pupping and also by seed. So they're able to reproduce before they die, essentially. So because this has caused such a major decline in both populations, there's been a lot of conservation work as well as monitoring work being done on both of these species, seeing how the populations are bouncing back or being hurt by these invasive weevils and how that's going. So in these in this work, it's really crucial to be able to tell seedlings apart, which isn't something that has been so easy in the past, being that they're so small and pretty hard to distinguish. So basically, the goal of my research was to find an accessible way to tell these two seedlings apart in the field. So to do that, I measured 70 seedlings from the Selby greenhouses. It ended up being 34 Utriculatas and 36 Fasciculatas. At least 5 from every representative age group that was available. And the measurements that I took were longest leaf length, height of the plant, a blade width of the longest leaf, a sheath width for representative plants that I was able to dissect, also a most horizontal leaf length, which is essentially kind of like a radius of a plant, and also like an entire diameter of the plant from widest leaf to widest leaf. So after that, I moved on to taking scanning electron microscope images of 2 representative plants from every age group for either species, both looking at the adaxial and abaxial sides of the leaves, so the top and the bottom surfaces. And I used ImageJ to analyze the entire diameter of one of the trichomes on the representative sides of the leaves, as well as the trichome wing diameter. So what I found from my measurements of the plants as a whole is that there's a higher blade width to longest leaf length ratio in Tillandsia fasciculatas in comparison to Utriculatas, which suggests that these plants have shorter and fatter leaves than the Utriculatas, which you can see pretty clearly in these images, and that these leaves look kind of more succulent and kind of shorter and fatter than the Utriculata on the other side, which has sort of slimmer leaves. On top of that, I also used my most horizontal leaf measurement, so like the radius with the plant height, to calculate an angle of the most horizontal leaf, which showed that Tillandsia utriculatas had a much lower angle overall than fasciculatas, suggesting that the growth pattern of utriculata seedlings is a lot wider and more horizontal than fasciculatas, which is also shown here. So you can see that plant kind of is growing in a more horizontal way than this, which seems to be more bunched together and growing more vertically. Oh, no. So the SEM images that I took provided some really interesting insights into differences in trichomes of the 2 species. And as you can see, the trichomes of Tillandsia fasciculata here appear to be a lot more ruffled and running into each other than the more uniform of Tillandsia utriculatas that kind of lay a lot flatter. And that was supported by the quantitative analysis that I did with ImageJ, which just showed that on average, Tillandsia fasciculata had a much higher average trichome wing length than Tillandsia utriculatas, which, as you can see here, they stick up a lot more and they stick out a lot more than the shorter trichome wings on Tillandsia utriculata. So, the goal of this project was to find the most accessible way to tell these 2 apart as seedlings in the field, and I feel like from my research, the best way to do that is by trichome density and the extent to which the trichome wings are ruffled on the 2 species, being that you don't need to make any sort of a calculation or intensive measurement in the field to be able to see this. Like these can be seen at lower magnifications, can be seen through a hand lens, and I feel like you would be able to, like, you don't have to compare them. Like, whereas my other measurements, it was like, well, one is wider than the other. I feel like you would be able to see a figure of a Tillandsia fasciculata seedling and be able to match it to what it is in the field. So, yeah, I just want to give thanks to Liz Gandy and Bruce Holst for allowing me access to the Selby greenhouses and helping me through this process, and also Brad Overly and Brian Sidoti for just like mentoring me and giving me these great opportunities, giving me rides to Selby, giving me good, great fieldwork opportunities all around. Also, Teresa Cooper for the original work that we went off of in a lot of our field work. Also, Lily Benitez for just, I feel like our work really complements each other's really well. Yeah. So thank you.